for this introduction. 1, 2. Thank you. Can you hear me? Yes, great. It's just wonderful to be here. I actually live in Venice too, Venice, California. And, but aside from the name, I don't think there's really anything that is related. And it's so just wonderful to be in this magical city again that I spent quite some time in as a child. Today, I like to give you some very broad comments, not about the present achievements of precision medicine, but again about what I think the future might hold and what the future challenges might be. And so, the first part of the title is just making medicine more personal, precise, and predictive. That's really the goal of precision medicine. And currently, the focus has been the collection, this incredible revolution of generating data sets of the genome, of the epigenome, of biomarkers. And that is what we call big data. And the question is this big data really sufficient to yield precision medicine? And so let me start with some, so I need to switch this on, all right. Let me start with some very simple points. Medicine is about people. Don't forget that. Medicine is about people, and yet we're all talking about and microseconds and seconds and minutes and hours and days. And so human health is phenomenally complex, billion-fold scale differences. How do we deal with this complexity? How can we imagine to make predictions based on genetic changes at this level about the health of people? How can we imagine doing that? Very complex. This is where the groundhog comes in. This is an American phenomenon. I don't think they exist in Italy. The groundhog is this strange animal that when on February 2nd, the day is cloudy or sunny, the groundhog comes out. And if the groundhog comes out, it will be an early spring. So based on that day, you can predict the weather 6 weeks later. This is a biomarker or a weather marker that allows us to make a prediction about what happens in the future. This is really what biomarkers are about. Biomarkers are individual measurements that sometime later down the line have predictive power that will predict what the disease outcome is or whether a drug works or does not work. And in order, now, the groundhog is really smart. And so another way to describe the groundhog in this case is that we need a statistical model to take these measurements in order to make a probability description about the disease. That's the basis of precision medicine, and that's how we're able to jump over these incredible complexities from molecules to people. In other words, this complexity from genes to organs is basically leapfrogged, is jumped over with a statistical model that we feed biomarkers and we make a diagnosis, we make a diagnostic decision or a therapy decision. That's the miracle of statistics. The miracle of statistics is that we do not need precise knowledge about everything that is in between, all of this complexity. And the miracle of statistics is that it goes across scales and is agnostic to causation, right? That is really what the power of statistics is. Some of these biomarkers work. They work really well. For example, a biomarker in this gene predicts whether blood cells are normal or when there's a mutant, are sickle cells, hemoglobin. This biomarker is 100% accurate. One gene, one outcome, one disease. Amazing. Sorry, yeah. So here we are. Other biomarkers are fairly accurate. We already heard about the breast cancer biomarkers BRCA1, BRCA2, where they give significant predictive power of whether a woman develops breast cancer or not, and how that treatment should take place. Very accurate, very useful. And one reason why these particular biomarkers are so useful is that we actually understand the causation. In the case of BRCA1, BRCA2, these are known repair proteins, and research has shown, molecular level research has shown, what these proteins do, and that they control DNA repair, and if they're missing, that cancer cells are prone to developing more mutations. Other biomarkers are not as accurate. They're still useful, but their predictive power is limited. Here are a set of biomarkers, and you see that in some cases, these probability curves of patients that have or don't have these biomarkers show differences in their survival or their response to therapy. And you see some differences here. So basically, if you look at these numbers, these are 50% or 45% versus 48% after some years. That makes a big difference. These are dozens and hundreds of people who benefit from these biomarkers, but certainly not everybody in these studies. And that is the reason for the limitation of these biomarkers, but first of all, that we don't understand fully what the causal relationship is, why they have the function that they have, and secondly, that diseases are simply that complex. And so combining multiple of these biomarkers turns out to improve predictive power. Rather than relying just on one, we can make measurements of 30 or 50 biomarkers, and we can find that even though when we look at these 30 biomarkers here for different tumor biopsies, there really is no relationship. But if we statistically rearrange that data, we find that it falls into some classes where at least some of the tumors show a very clear distinction that when these genes are in one state, the green state, or in the red state, that they segregate, and that segregation results in different survival curves. And so that type of recognition, that was recognized about 15 years ago, that combining different biomarkers together allows more predictive power. And so these statistical models, of course, also become more complex. Now, take this forward, instead of just doing measurements on 30 genes, we can have measurements on thousands. We start with the genome, we have thousands and millions of different biomarkers. We can relate these biomarkers to clinical records from thousands and millions of different people. Our statistical model will become more complex and more predictive about a new patient who presents these particular biomarkers. And that will allow us to improve diagnosis, personalized therapy, and reduce costs, the basic tenet of precision medicine. So what kind of data do we have? Well, we have genomes, we have epigenomes, we have transcriptomes, we have proteomes, we have clinical records, we have metabolomes, we have microbiomes, as we heard, and all together, we bring them together to not only improve diagnosis, personalized therapy, reduce costs. we can identify patient cohorts for other studies, and of course, resurrect some drugs that are not good for everybody, but we can identify a specific set of patients for which this drug is beneficial. And then the next step, other than precision health or precision treatments, is precision health care. Health care is not about disease care. Health care occurs prior to becoming sick. And for that, we need another set of data, the data that exists before we become sick, which is really about lifestyle data, environmental data, and this is where the health monitors, the wristbands, the iWatches, the mobile health is so important. And again, these data are put together in a way through statistical models that don't require precise knowledge. That's the excitement. That's the holy grail. And so, will that be possible? Predictive power of statistical models is, of course, a function of the amount and quality of the training data. How much data do we need? We have the genome. We differ by about 10 million SNPs. We have the environment, the lifestyle. All of these are differences between people. In the case of, here we have 10 million SNPs. We have millions of variants here, millions of variants here. And if you combine these numbers, we get into very, very large numbers because these SNPs, of course, don't function individually, they function together. And the SNPs function together with lifestyle and with environment. These are all multiplications. And so the net result is actually, gazillion, the number that the calculator can't compute. Sorry. In fact, so this number is so large that that we require more clinical records, not just 100,000 genomes, not just 1 million in the NIH. We require more clinical records and more molecular data from people than there will ever be people on this planet. It's a very depressing thought. Very depressing thought. Is there a way out? This is where the plane comes in. Is there an alternative approach? Well, this is a very complex machine. A Boeing 737, that's what the web tells me, is 367,000 parts. I think there must be a lot more than that, but somehow that's the number I'm getting. And it's a complex way of assembling a plane. It relates to the complexity of humans by maybe several orders of magnitude. But the point is that this plane, despite its complexity, is extremely precise. It's the safest mode of transport. In fact, it is so predictable that we can simulate how to fly an airplane. And every time we simulate, it's exactly the same answer. How can that be possible for something that's so complex that it can be so predictable? Well, the reason is it's not based on statistics. It is based on physical principles, and these physical principles are described in mathematical equations. For example, the Bernoulli equation that relates the speed of a wing with the force, the lift that carries the airplane. And so that is a different kind of math. Let me explain. Statistics is the ability to have a probability of two events occurring that predicts a third event, for example, or the probability of many events that determine C. the mechanistic way of describing, of relating events is algebra, or differential equations, or it's a different kind of math. And this is precise. It has an equal sign. The power of statistics is that it's agnostic of knowledge, mechanism of causation, and scale. This is precise, but it requires a lot of knowledge. So what I'm saying is that we should not be satisfied with a statistical model that takes us from genes to humans, but that we embrace the complexity and we build mechanistic models of each of these scales for the DNA, for the networks, for the cells interacting, for the organ systems. And we link them in a multi-scale mechanistic framework to make predictions that are precise, that are non-statistical. So how do we develop mechanistic models? First, we have to identify all the components and develop the parts list. Actually, we have the technology to do that. We have many of the components, most components. We have to learn the physical principles that connect these. Sorry, we have to learn the physical principles that connect them. We need to learn how each part works in the context of the system, and we have to build a computational model. And then we have the required knowledge to simulate physiological functions. The great news is there's been a huge amount of investment in generating precisely this kind of knowledge. Again, we're referring to the NIH has invested a very large sum of money over the last few years in generating knowledge. And that has given rise to, essentially, the academic departments. The academic departments at every university of molecular biology, biochemistry, cell biology, and physiology that generate knowledge at each of these scales. Precision medicine, focused solely on statistics, does not utilize this knowledge. That knowledge that has been accumulated in each of these departments. And so, what kind of knowledge exists? We have knowledge about gene functions, we have knowledge about enzymatic constants, we have knowledge about drug interactions, we have knowledge about the molecular networks and how they work. We have knowledge about where proteins localize in the cell and how they move. We have knowledge about feedback within each of these systems that is so important for regulation. And of course, we have knowledge about the redundancies of proteins or how they synergistically work. The problem is, how do we extract this knowledge? Because then currently the knowledge is present in scientific papers of this sort and is not easily extracted. So we need to extract this knowledge to develop regulatory networks of this kind. This is work that actually was done in my laboratory some time ago. We can take this paper, this scientific paper, and describe it as a mathematical equation. And when we have a mathematical equation, we can do simulations of what happens in response to a pathogen infection, for example. And here's the data, and here's the simulation. We can compare simulations with data to assess the quality of our models. So this is hard. This is hard. It takes a lot of work. Is it necessary? And here I give you 2 or 3 examples of why it is necessary to have models that are based on mechanism. Consider Newton. Newton measured how long it took for an apple to fall from a tree and measured the time. And he did this for an apple that fell from this branch or from that branch, or from this branch, or from this branch, or from this branch, and he got data points. And his measurements were good, but they were not perfect. And so the numbers are a little bit scattered. And then any normal precision medicine person would have said, oh, I can see a correlation, and I draw a line. And the R-squared value is acceptable, 0.9, 0.86. The correlation fits well. Newton was not satisfied with this. Newton said, I want to understand the physical principles, what really is going on. And what he discovered, that in fact, the time is a function of the height, but of the root to the height. And the actual curve is this one. It fits just as well, but it's the right curve. Now, does it matter? It does matter if you want to predict how long it takes for an apple to fall from the Tower of Pisa, or for an apple, or for you to shoot a rocket to the moon. Because you can see very clearly that those curves diverge, and if you go to the moon, you'll be probably somewhere completely different. You'll miss the moon by a long shot if you try to base your rocket engineering on a straight line. So the knowledge-based models allow you for predictions beyond the training range. Very simple illustration of that. And it's absolutely true in medicine too. If we want to make predictions that are outside what we've trained our models to be, we need to have knowledge. We can't do it based on statistics. Here's another example. Here's a set of data points. This is a biomarker. They scatter. Who's the person who's sick? Or who will be sick in 6 weeks' time? You tell me. Is it the one at the top, or is it the one at the bottom? Neither. It's this one. Why? Well, the underlying mechanism actually shows that over time, for each person, there is a biorhythm. That marker goes up, goes down at different days, or throughout the course of the day. And here's a person where that biomarker does not go up or down. It stays the same. And that is the problem. That is the reason why that person is going to develop a disease, or why the drug is not going to work. So if you have this kind of measurement, you completely miss what this biomarker should be telling you. And having that knowledge will allow you to develop a time series of measurements at precise time points so you can interpret the data. Knowledge-based models interpret confusingly complex data. Similarly, the data for a biomarker might be looking like this, and if you inhibit the biomarker with a drug, that drug might turn this thing down, might reduce the scope, or it might absolutely prolong this. So drug interactions with the biological system, the consequence of a drug interacting with a system are dictated by nonlinear relationships that you cannot predict unless you have knowledge of how molecules are connected with each other and how precisely the network works. So what does that mean? It means that in order to put all of these data sets together, in order to put all these data sets together, we can't just use statistics. We need to think about what kind of algorithms we need to develop. And we need to develop knowledge. And so the multi-scale modeling requires statistical genetics, genomics, databases, networks, dynamical systems, structural informatics, all kinds of algorithmic approaches need to be integrated, and the right one needs to be picked to develop models. And so that's precisely what the interdisciplinarity that was addressed earlier in this conference means that life sciences and health science, medical school and engineering have to function together. And this is kind of what we've done at UCLA. These are all professors of various disciplines, from computer science to physical sciences to mathematics to life sciences and neurosciences and clinicians, to come together to develop models for precision medicine. And so the institute is called Quantitative and Computational Biosciences, QC Bio. QC sometimes stands for quality control, and we like that. We develop models for big data, and we extract knowledge to address the big knowledge, to leverage the big knowledge in the literature, to develop models that benefit predictive biosciences as well as precision medicine. And so we focus on research training, and education. And so in my lab, we develop models for the immune system. And so in recent work, which I will not talk about in detail, we have developed a mechanistic model for the molecular network as well as for the cellular network to develop a model for immune responses. If we take molecular data, cellular data from movies to understand how the population of immune cells works. And so you already saw the last slide. This is my last slide. Max and my group do such work, and I invite you to join us. Thank you very much.